check it out. It's kind of interesting. We'll hear, well, we heard about rainfall. We're going to hear, and we've heard about silver swords, so check it out. Actually, we haven't heard about rainfall yet, have we? We will be hearing about rainfall. Check it out. I'm going to get going. Josh, can you hear me in the back? Okay, I'm going to speak from here. I'm not going to cough this whole lecture. Coral reefs, my favorite topic. So you just got to stop me at 20 after. We're going to be here till 20 after, I promise, because this is obviously my favorite topic, and I'll get carried away, but I'll do my best to stay on track. Before I get going, any questions or problems with where we are right now? Everybody finished quiz 4, right? Turn it in? Yep. Okay, good. If you have any issues, questions, you need to talk with Josh or Dr. Ahmed or me. Don't let it get any further. And you should be just about finished with your project, right? Right? You should be starting with your project anyway. That's the point. Don't let it get to the end of the semester because everything else stacks up. Uh-oh. Okay, keep going. So coral reefs, I mentioned them last time as one of the kinds of biogenic ecosystems that we see around the world. Coral reefs, mangroves, and seagrass beds. Ecosystems structurally built from living organisms. Here's a typical Hawaiian coral reef. And we can tell it because actually in Hawaii, about a third of the species of corals that we have are endemic. You know what that is? So if I see Paredes compressa finger coral in a photograph, I know, hey, that must be Hawaii. If I see fire coral, nasty fire coral, or many other species of corals that are common throughout the rest of the Pacific, I know that's not a Hawaiian reef. This is also a Hawaiian reef, but if you're a snorkeler or a diver in the main Hawaiian Islands, this is not an image that you see regularly. Anybody in here been up to the Northwesterns yet? Cool, put it on your wish list. I've only been about halfway up with a group of teachers, but it's a different universe. Should I dim the lights a little bit?
Yes.
Go to sleep. So, coral reefs are biogenic, and the energy that they use to build their skeletons and what makes a hard coral reef is derived from the sun. And it's because of the little tiny, literally tiny, single-cell algae that live inside coral cells that this can happen. It's miraculous. These little cells look like this. They're plain, simple, as I said, tiny. These are dinoflagellates of the genus Symbiodinium. And they do what all good plants do. That is, they photosynthesize, taking energy from the sun. Oops, go back one. And turn it into carbohydrates, which they share with their coral hosts. As part of this process, or linked to this process, is calcification. Corals are able to take carbonate and calcium ions out of seawater and deposit it as calcium carbonate, the hard part of a coral reef. So these are representatives of common corals in Hawaii, and what's at their core are these little guys, the zooxanthellae. These are the power packs. Corals, there are corals that don't have zooxanthellae, but they don't make coral reefs. They'll make a skeleton, but they don't have enough extra energy to lay down that calcium carbonate skeleton. Another big important photosynthetic group on coral reefs are the Hawaiian seaweeds. I'll just let that trickle through. Come on. And in Hawaii, these are known as limu. Very important to Hawaiian culture because many species are edible. And we had an amazing woman here, Dr. Isabella Abbott, first Hawaiian PhD in science, who studied Hawaiian seaweeds, literally wrote the book. So here in the middle of the Pacific, we have one of the best records of the diversity, the biodiversity of limu, of seaweeds on coral reefs of any place in the world. They come in brown, green, red groups, and they're tremendously abundant and important. So photosynthesis is put on by two really different shows. The limu that I've just shown you here, the Hawaiian seaweeds, and the photosynthetic algae, the dinoflagellates that live inside a coral cell. One thing that makes coral reefs really interesting and cool is their high level of productivity. Their primary production, so that's the photosynthetic production on a reef, done by the plants themselves, is over 500 million tons per year on a global basis. I'm sorry, primary production around the world is 0.56 billion tons per year. Coral reefs, primary productivity is about 650 grams per meter square per year. And if you multiply that, I'm sorry, I'm over here on the side, I meant 2,000. 650 is cultivated land. 2,000 grams of carbon per meter square per year. And if you multiply that by the total area around the world that coral reefs make up, about 17 million square kilometers, do the math and you come out with about 11 billion tons of carbon fixed per year just by coral reefs. So I want to point out here that coral reef primary productivity ranks high among many other ecosystems, about the same as tropical rainforests. But coral reefs around the world don't represent as big of an area as some of the other ecosystems. Open ocean being the dominant area. Yes, question?
You said 11 tons. I think that's for cultivated land. Coral reefs is 0.56.
I did. 1, 2, 3. Did I get that wrong? 1,
Yeah.
2, 3. 0.28, even smaller. Thank you for pointing that out. Coral reef area, everything looks slanted and long from here. I apologize. Coral reef area is even smaller. 0.28 square kilometers. 0.28 million kilometers square. So this productivity is important and high. Coral reef, global marine fisheries around the world, all marine fisheries represent 100 million metric tons per year, and those that come from coral reefs, about 9 million metric tons per year. So 0.9% of all fisheries productivity and 0.09% of the area of the planet is that coral reefs have about 100 times on average the productivity of any other area on land. So they're tremendous fishery stocks, but they're also tremendously vulnerable. We'll talk about that the last week of class. And they're very complex because coral reefs are living organisms, living animals, based on living animals and plants, and they respond to nutrients like fertilizers, phosphate and nitrate, to currents, to light, and to water motion. And I'll tell you how. So, and primary productivity on reefs is very high. There's a lot of carbon being fixed, but the export of that productivity, net productivity of coral reefs is fairly low, about 1%. And that's because there are many trophic levels on coral reefs. The nutrients on coral reefs are tightly recycled, and there are many species interactions, both competition and predation. So the title of this talk, of this section, is Biogenic Ecosystems in an Oceanic Desert. So a little island chain, like we're sitting on, the Hawaiian Island archipelago, in the middle of the Pacific, there are not a lot of nutrients in our surrounding waters. It's pretty much an oceanic desert in regard to nutrients. Closer to continents, there's a whole lot more nutrients that enter the coastal ecosystems from land, but not out in the middle of the ocean where coral reefs thrive. So I mentioned that corals are animals. They're related to two maybe more common organisms, anemones and jellyfish. All of these animals are very simple. They're basically a mouth surrounded by a ring of tentacles, and the mouth leads to a blind sac. So this is what a little anemone looks like. A jellyfish is about the same thing. Turn it upside down. It's a mouth surrounded by a ring of tentacles. The mouth leads to a blind sac. Very simple, simple organisms. Only two body layers, only an inside and an outside. Anemones, jellies, and corals are all united by one really interesting type of cell. This is a cnidocyte. There's the spelling here. The C, C is silent. Cnidocytes are stinging cells. Anybody ever run into a jellyfish with stinging cells? An anemone with stinging cells? Sometimes they can sting you too.
What's an anemone?
Thankfully, the corals in Hawaii, although they have stinging cells, they don't penetrate our skin. You might get a rash or an irritation, but it won't double you over in pain like a fire coral will, that are very common on reefs in the Caribbean and other places in the Pacific. Yay, Hawaii doesn't have fire coral. Question?
What's an anemone again?
What is an anemone?
Yeah, difference between an anemone and a jellyfish.
Here's an anemone, a graphic of a sea anemone, and this is a graphic of a jellyfish. They can be very small and they can be this big around, common members of the intertidal.
Oh, so an anemone is basically one of those, like they stay in one area type of sea plant or animal, yeah?
It's known as anemones are the polyp form of this kind of organism, of these cnidarians. This is the cnidarian body form. I can show you pictures of anemones after class.
Okay, thank you.
And all of the jellyfish, anemones, and corals are united in that they have cnidocytes, these stinging cells that are released either by pressure or by chemicals in the water. And they can cause an irritation to severe pain. And some jellyfish stings can even be lethal. You really don't want to tangle with jellyfish because their stinging cells have venom attached and can really hurt. This is a close-up of what a coral looks like. Been talking about coral skeleton, calcium carbonate skeleton. That's shown here in the white. The coral animal itself, it's like an anemone, but most corals are colonial. So here's that mouth surrounded by a ring of tentacles, and that mouth leads into a blind gut. The outside is shown here in green, the inside shown here in red, and the coral actually secretes the calcium carbonate skeleton. It takes carbonate and calcium ions from the seawater again and deposits them as skeleton. Usually it's pretty slow going, this deposition. A big lobe coral in Hawaii, for example, can only grow about a centimeter a year. Pretty slow. Some of the fastest growing corals, some of the table corals, the acropora, say off of in Indonesia, can grow 25 centimeters a year. A branch can grow that much in a year, about a foot. So quite a bit faster. But corals take their time in depositing this calcium carbonate skeleton. And the reason that they can do that is because they have zooxanthellae inside their cells. This is a close-up of the surface of a coral. Can you see that it's a colonial animal, lots of anemone-like polyps across the surface. See the mouths surrounded by a ring of tentacles.
Also, those are sea
And if I was a little, hold on, we'll talk after. If I was a little fish swimming up to this coral and getting ready to take a nibble, the coral's polyps actually pull into the skeleton where they're protected from the mouth of the fish. So what does this tell you? Does a coral have a brain? Not so much. Let me go back here. No brain, but it does have a loose nerve network. So that a polyp that's bothered over here is loosely connected through its nerves to a polyp maybe over here. So a fish bite over here will be sensed by the colony, and these little guys pull inside quickly. Let me go back here again. So no brain, no heart, no lungs, no kidney, no organs. Just an inside and an outside, a very simple organism. That's going to become, it is important, and I'll explain another aspect of that in a minute. And corals can grow in a variety of shapes and sizes. From a single polyp here, this is called a mushroom coral, to a big, massive coral up here, branching, plate-like. I told you I wouldn't. But in Hawaii, we have about 60 species, 60 different species of corals. How can I say about 60 species? I'm a coral expert. I'll just say it. I am. But why don't I know exactly how many species of corals there are? Shouldn't I? It just depends on who you ask. Some people really split the species of corals into numerous groups that they call species. Others lump them together and just say, no, those aren't different enough to really be a species. How do you define a species?
Well, there's different ways.
What's that?
There's different ways, but generally
ways.
reproductive, like if they can reproduce together.
Potentially reproducing individuals, right? Potentially reproducing individuals. Shouldn't we be able to know if corals are potentially reproducing? Well, here's what a coral looks like when it's actually reproducing. I showed you coral larvae among the first couple of weeks of this class. These are coral eggs and sperm being released from the polyps. These bundles have inside them about 8 to 12 eggs and a bunch of sperm in the middle. Check that out. So they're released, and eggs are mostly lipid. So do you think eggs sink or float? Sink or float?
Float.
Float. They float. They're fat. They float. So that most coral eggs float toward the surface. This is in a coral that has eggs and sperm in the same polyp. Is that wild? We call that hermaphroditic. This is a coral. This is actually finger coral where the sexes are separate. It's called gonochoric. Separate sexes. One colony over here is male, one colony over here is female. And this is a rare shot of a finger coral releasing sperm. And a colleague of mine caught this coral in the act, and it was no easy trick. Do you know when they do it? Between about 11 o'clock at night and 1 o'clock in the morning in July and August. This species right after full moon. So you get 2 shots at it. You better be there on the reef or in the lab watching these individual colonies spawn at just the right time. So back to the question of, are these species interbreeding? Are these organisms interbreeding and thereby all the same species? It's hard to tell because you really have to work hard to know when they're spawning. And guess what? They're shy. So you get them all ready. You might bring a colony into the lab so that you can comfortably watch it all night long. It's hard to be in the water hovering over a coral colony from like 10 p.m. until 2 a.m. just hoping that it's going to spawn. Sure enough, if you're watching, they won't do it. They're not really shy because remember, they don't have a brain. But these are really hard observations to make, is my point. However, the Australians, actually grad students in Australia in the mid-1980s, you know, Australians have a reputation as being a rambunctious group, and these grad students certainly were. They were willing to stay up overnight to see when the corals were spawning. And on the Great Barrier Reef, they found that most of the coral colonies there all spawn on 1 or 2 nights after full moon in springtime, and most of them produce egg sperm bundles. So the next day, if you were flying over the Great Barrier Reef, you might see slicks that look like this. These are slicks of coral eggs, and now developing embryos at the surface the night after this mass spawning event. Those grad students made big news when they made this discovery, and a few of them came up to Hawaii the next year to help us here, scientists here, learn how our Hawaiian corals reproduce. Do our Hawaiian corals do it the same way?
Nope.
This is another great photo a student shared with me a couple of years ago. She was doing work at Johnston Atoll, and the next morning, they didn't actually know what had happened. They got up the next morning and saw a pink slick washing up on the shoreline. They couldn't imagine what it was, so they took a sample, put it under the microscope in the lab. They happened to be there with a microscope, and sure enough, they found out that these were coral egg sperm bundles. The egg sperm bundles begin to break up. If most of the spawning is done in Australia, for example, at 8 p.m., the coral egg sperm bundles begin to break up in the next couple of hours. And you can see the eggs clearly, fairly clearly here, and see this inside? That's the sperm packet being released. So, like most animals, crossing within an individual is avoided. So these sperm need to fertilize eggs from another individual, which makes it kind of cool that they're all spawning at just about the same time. About 8 o'clock at night, springtime, just after full moon. Most of the species spawn the same time, and so most of the individual coral colonies do too. So these sperm can start looking for, hopefully, a neighbor to fertilize. And after a few days, this is what a coral larva looks like. Is that not adorable? It looks like a little smurf. There's nothing very special about it. It's got a front end and a rear end, and it kind of snuffles around on the bottom until it finds a good place to live where there's no other corals living there. Nothing else is going to eat it. It plunks itself down, and here is a brand new 1 polyp coral colony just getting ready to go. This little guy actually settled down on a piece of rock on a bowl on the top of my desk out at Coconut Island, which was very fortunate for me, so I could see what a brand new coral colony looks like. This is just getting started. So here's that life cycle. It's really actually simple. Here's an adult coral colony releasing either egg sperm bundles, or in some species, either eggs or sperm. Fertilization happens in the water column. Development begins. A larva is formed. It plops itself down in a good place on the reef and begins to divide and grow into another adult colony. And from the time it settles down here to the time that it's reproductive, for most corals takes about 2, 4, 5, up to 10 years. Another hard number to get, because you have to be watching that same coral colony from the time it settles down to the time that it reproduces, which means you got to hang around one place and be very attentive for a long period of time. I mentioned earlier that corals respond to a number of influences. The major ones, I mentioned nutrients. Other major ones are light, water motion, and water quality. So here's light. Why do corals respond to light?
Photosynthesis.
Photosynthesis, because of the algae that live inside their cells that enable them to calcify. They live in the photic zone, which is defined kind of arbitrarily as about 100 meters, about 300 feet deep in the ocean. Is it always 300 feet deep no matter where you are in the ocean? Heck no. Some areas are much more turbid than others. Some areas the water's very clear. I'm not saying that corals don't exist deeper than 300 feet in the ocean. They do. They just don't grow very quickly. Optimal growth occurs at about between 5 and 15 meters on a reef. So between 15 feet and 50 feet on a reef. That's optimal area for growth. That's where the light is just about right. Any shallower, and there's too much UV. Any deeper, and there's not enough light to drive calcification. And again, that's really dependent on where you find yourself as that little coral colony. If you're that little coral planula swimming around and you swim under a ledge and plop down, it might be a safe spot, but you don't get enough light. It also makes you, but if you settle on the top here, right here on the top, good spot. But it also makes you very vulnerable to the next little fish looking for a delectable little bite. So it's tough being a little larva and figuring out the best place to settle. Most corals, it turns out now that we've been looking for them all this time, they like to settle in cracks and crevices. So when they're first settling down there, they're protected from too much UV, and then they begin to grow out of that little crack or crevice slowly. But for the first couple of months that they're there, they're really hard to see. Here's just a graphic that reminds us about how light falls off in the ocean. Here's at the surface, 100% of surface light. And as you get deeper on the reef, this is in meters, 15 meters, 30 meters, light is less than 1% of what it is at the surface. So it attenuates. Light decreases with depth. Not only does light, in general, it decrease with depth, it decreases preferentially. The longer wavelengths, the red wavelengths, less energy are absorbed closer to the surface. So red light doesn't penetrate all the way down to 100 meters, for example. It gets cut off mostly before, well, again, it depends on where you are, how much stuff is in the water. But if you're a diver and you're, say, diving at 20 meters down at 60 feet and you cut yourself, have you ever done that? It is weird. Your blood looks green because the red light doesn't penetrate down that far, and you know you're cut, you know you're bleeding, but it's green. Red light is cut out preferentially. Coral reefs actually thrive in areas of water motion. There are some great surf sites that are right on top of coral reefs. I believe this one is in Indonesia. I've snorkeled over coral reefs in Fiji that are this deep below the surface at a great point break. Why would the corals thrive where there's this much water motion?
Because
they can't move around. They're stuck on the bottom. From the time that little larva settles down, that's where it's going to live or die its entire life. So that means that everything that falls on it stays on it because it can't get up and move or shake it off. So they're stuck. So it depends on water motion to bring in new oxygen and take away waste products of carbon dioxide. So they thrive in areas with high water motion. And you can see this from the air when you fly over some interesting atolls in the ocean. The windward side, where there's more water motion, will be more highly developed than the leeward sides. Corals like water motion. Corals hate fresh water. They do not grow where there are stream mouths. Why would a coral hate fresh water? We mentioned that they don't have true organs. They don't have kidneys, so they can't deal with fluctuations in salinity. That's what our kidneys do. That's what a fish's kidneys do, is help them deal with changes in salinity. So where salinity is reduced, like where fresh water comes out of a stream, look what happens. The reef just ends here, and begins again where there's enough mixing to raise the salinity. Corals hate fresh water. There was a guy that we keep talking about in this class. It's like we think he's a hero or something. His name is Chuck. Charles Darwin was on the Beagle, a cruise that lasted for 5 years, 1831 to 1836. Look at the trek he took. Can you imagine being gone from his wife and, did he have 7 or 9 kids, something like that? For 5 years, he saw some interesting parts of the world. We know he's famous for what he saw and registered in the, where? Galapagos. But he saw a lot of other interesting places, and he made tremendous observations. He saw, particularly in French Polynesia, fringing reefs and patch reefs and barrier reefs and islands like this, little atolls. Now he didn't see them from the air, right? He sailed by them on a ship, taking notes the whole time. Here's what he might have seen in Tahiti. Again, he was sailing by on a ship. A fringing reef and a barrier reef and a lagoon. Similarly, a fringing reef. And he started thinking about it. How did those little atolls form? Those circular or triangular or square atolls in the middle of the ocean. How the heck could those have formed? And he came up with this really interesting idea, a hypothesis, a theory, a subsidence theory, and you've heard this before in this class. His idea was that corals grow around volcanoes, on the edge of volcanoes. Volcanoes weigh a lot and they sink, but as they do, the corals around their edge keeps growing up. So pretty soon you have a barrier reef out here with a lagoon in the middle and still fringing reef closer to shore. And pretty soon the volcano's gone and all you have left is that barrier reef. No trace of the basalt volcano is left. And he called this an atoll. I don't think he actually coined the term atoll. Here's another view, I like this one. Volcano, fringing reef growing, volcano starts to sink, fringing reef keeps growing up, volcano's sunk and you can't see it from the top anymore. That's why this is a hypothesis. He couldn't see that this had happened, but he put it together that this was the most likely idea. The alternative hypothesis was that there are bumps in the bottom of the sea. Sediment accumulates on these bumps until they get closer and closer to the surface, and they're finally grown up into the photic zone. When they're in the light, then corals can settle and grow on them, and then corals begin to grow up. Chuck Darwin put out this idea. I love this quote. In a letter to Agassiz in 1881, less than a year before he died, Charles Darwin said, "If I'm wrong, the sooner I'm knocked on the head and annihilated, so much the better. It still seems to me a marvelous thing that there should not have been much and long continued subsidence in the beds of the great oceans. I wish that some doubly rich millionaire would take it into his head to have borings made in some of the Pacific and Indian atolls and bring home cores for slicing from a depth of about 500 or 600 feet. This was a big wish in Darwin's day, in the late 1800s. But there were some people in the mid-next century, in the 1950s, that were doubly rich millionaires, and they decided to test this hypothesis. These doubly rich millionaires were actually the US government, and they were working on atolls like Bikini Atoll and Inuiatok in the middle of the Pacific, and they realized that they could do what Darwin had wished for so much, and that is drill down 500 or 600 meters deep to see if there was sediment all the way down or carbonate, coral calcium carbonate over the basalt. Question?
Bikini Atoll, isn't that where they tested one of their nuclear bombs?
Pardon me?
Isn't that where they tested one of their nuclear bombs?
This is where they were testing nuclear bombs, and that's exactly why the US government was there. Good question. And they had lots of capability when they were there to drill. So they drilled at Funafuti, at Inuiatok, and at Midway. Here's Eniwetok Atoll, perfect place to test Darwin's theory of atoll formation. They did the drilling at Funafuti, and they drilled and they drilled and they drilled and they drilled, and they only found limestone, only calcium carbonate. Huh. Didn't really answer the question. At Eniwetok, they drilled 5 times deeper. They drilled and drilled and drilled through calcium carbonate reef, and guess what they finally hit? Basalt. They finally hit that volcano. Question answered. There were no sediments. Darwin got it right. One of the many things he got right, and one of the many important contributions he made to how we understand this planet. What Darwin didn't see could have answered his questions long before, actually. He never saw the Hawaiian Islands. Too bad. He didn't see the terrestrial side or the marine side. Because we have, throughout this Hawaiian chain, fringing reefs, barrier reefs, lagoons, and atolls. This is French Frigate Shoals in the northwestern Hawaiian Islands. This is Kure Atoll, way up at the top of the archipelago, 28 degrees north. And there are also remnant basalt in the Hawaiian archipelago where he could have put it together. Aha! He could have seen that here is a trace of a volcano still above the surface and coral reef growing around the edges. This doesn't happen in most of the Indian and Pacific Oceans. There's no trace of the basalt formation left. But in Hawaii there is. Quickly, I want to go over some atoll morphology. Just a couple of words that I'd like for you to have in your vocabulary. Here's a good one, motu. It means island. It's a little island. So here's the edge of an atoll, and you can see these little islands around. Here's another close-up, an aerial view of an atoll. Here's the reef flat, the windward side, and over here the reef is going to be growing faster or slower than on the opposite side?
Faster.
Faster, because of water motion. Here's that lagoon in the middle, and here are little motus, little islands that are vegetated. That's what it means, a little vegetated island. Another example, the reef crest, reef flat, and here's an islet. Might have palm trees on it, but vegetation. We've been talking about water motion in the windward and leeward sides of islands. When here comes a big swell, going to turn into a wave as it hits an island or an atoll in the middle of the Pacific or wherever it is, might be the North Shore here. It hits, and guess what? The reef itself disperses the wave energy into what we call spur and groove morphology. So when a wave hits an island, it's not just a wall of coral. It's actually spur and groove, hills and valleys, hills and valleys, hills and valleys. One thing that really makes it fun to dive on the outside of a coral reef, although it's really sharky out there too usually. It's a lot more comfortable and safe inside the lagoon. But if you're brave enough to go over the edge to the outside, you'll see these buttresses, these spurs are the hills and the grooves are the valleys, and they break up the wave energy. One of my favorite places in the world, this is a whole other lecture, but I'd love to talk about it sometime. This is the tiniest atoll in the world. It's in American Samoa, part of the US. It's a US Fish and Wildlife Protectorate. Tiny, in the shape of a little diamond. And it's called Rose Atoll, not just because it's kind of pink when you see it from the air, but the first captain who got there actually named it after his wife, whose name was Rose. That's true. Quickly, I want to go through succession. Succession is when you have bare nothing and brand new earth, and plants and animals move into it and begin to start an ecosystem. There are primary colonizers. The next group of species that moves in are competitors, and then it reaches what we call a climax stage. And you may have studied about this in terrestrial ecosystems, where as time passes, the ecosystem changes from one that's dominated by grasses to shrubs, trees and pines, and maybe a hardwood forest is the climax community on land. In the ocean, it's a little bit different, and we have wonderful examples of this in Hawaii. When do you see brand new land underwater in Hawaii? When lava flows into the ocean, right? Brand new, nothing's living there at all. It's been really, really hot molten lava. It hits the water and it cools, and then what happens? There's a great paper, and it's in your readings. Take a look at it by Ricky Gregg, who was an oceanographer here at UH, and Jim Maragos, also oceanographer, and he literally wrote the book on reef corals in Hawaii. And they, way back when, Jim Maragos was a grad student here. He actually lived in a shipping container on the roof of this building, HIG, because he was this poor graduate student. Not so much anymore. But they wrote this great paper on the recolonization of hermitopic corals on submerged lava flows in Hawaii. So they could see where the flows got into the water, and they knew the ages of these lava flows. They were able to write a really interesting paper. So here's what lava flows look like when they hit the water. We have some of these on some of the videos that you'll watch this semester. These guys were crazy. This photo was taken by Rick Grigg. I think this is probably a double hose regulator back in the 1970s. They had a spear, but they could swim right up to molten lava underwater. Can you imagine? Don't put your hand in there. So this is fire hot, molten lava hot. But then they were able to look at these lava flows of different ages and make some very important observations. Initial colonization, just by mostly bacteria, microbes to start with, begins in the almost instantly. But in the first 3 months, you can start to see a fuzz of microbes, blue-green algae, living on the bare lava, what used to be bare lava. Over the next 10 years, those little coral larvae, the little planula, begin to find it. And since there's nothing else there, it's a pretty good spot to settle down in. And there's lots of small holes, lots of small pukas in new basalt, so a good place to settle down. So on this 10-year-old lava flow are lots of, we call it cauliflower coral, Postlepora meandrina, and they're all about this big. So that's how fast the cauliflower coral can grow in 10 years. Longer than that, in areas where they mapped and studied lava flows that were about 40 years old, they started to see something different take place. Corals were growing like crazy, so much so that they were growing right next to each other and competing for space. Some corals are better competitors than other corals, so the ones that could grow fastest, the branching corals, actually outcompeted some of the corals that weren't as persistent, weren't as fast growing. 100-year lava flow becomes dominated, in this case in Hawaii, by our endemic finger coral. It's the fastest growing and the most persistent on our coral reefs. Briefly, I wanted to run through species diversity. How many corals about occur in Hawaii? About 60, somewhere between 55 and 75, let's say. This is how many species of coral occur around the world. This was put together by Charlie Barron, who wrote the book, Corals of the World. A new paper just came out last year in which he was able to plot the number of species from less than 50 to up to 600 in particular areas. So look at where the dark area is right here, north of Australia, near Indonesia. This is what we call the coral triangle. It is the center of biodiversity for corals. Up to 600 corals live on any particular reef. Aren't you glad you're not studying coral reef ecology in the coral triangle? You'd have your hands full just learning the species. I want to compare for you and give you some idea of the level of endemism on land. So we've been talking a lot about species in terrestrial ecosystems. These are the number of terrestrial genera, number of terrestrial species in Hawaii, compared to the number of marine genera, which is similar, but look how much fewer species, endemic species there are in marine ecosystems in Hawaii than in terrestrial ecosystems. Why should that be? If you look across all of the groups of invertebrates on reefs, from sponges through crabs and shrimp, snails and bivalves, echinoderm, sea urchins, and little worms. And then here's this group, the one that we've been focused on today, the Cnidaria. The total number of species shown in blue, and the number of endemic species shown in red. Look at this total number of species here. Super diverse. And the number of endemic species also high. In this group, the gastropods. Guess what? It turns out that we also, besides the person who was the expert on Pacific seaweeds lived in Hawaii, the person who was the expert on Pacific gastropods, Dr. Allison Kay, lived in Hawaii. She's the person who got this class started way back when, and who actually wrote the book. Is it the same level of diversity all through the Hawaiian chain? I want you to think about that. Do you suppose that the biodiversity is the same on the Big Island, brand new lava flows, as it is up at Curie at the north end of the chain? No, of course not. Wouldn't be interesting if it was. But what I want to point out is that, I'm going to skip to this one, I'm running out of time. Most of the recruitment that happens throughout the chain comes from adults on the same island. Isn't that interesting? These are data that have just come out of the Brian Bowen and Rob Tonin lab at Coconut Island over the last 10 or 15 years. Lots and lots of papers, and they've looked at, oh gosh, over 100 different species, and they see that this pattern is pretty standard. I'm gonna go back to this one. There is dispersal between the islands, but there are also these divisions that kind of, there's not as much dispersal between the north end of the chain, the south end of the chain, and then in the main 8, there are also divisions between Kauai and Oahu, between Oahu and Maui Nui, between Maui Nui and the Big Island. Interesting, whole new area of study. Okay, oops, come back. This is my end. The things I want you to take away. Yeah. There we go. Almost all of the marine species in Hawaii come from the area of the Coral Triangle, no matter what group they come from. There are fewer species as you go west and toward the area of highest diversity in the Coral Triangle. Many, but not, certainly not all Hawaiian marine organisms come from groups that typically have long-lived planktonic larvae. Endemism in marine species is primarily at the species level. Endemism can be really high, but one of the new studies that just came out in the last few weeks is that from our divers here working up at Morrow Reef, down at 300 feet, did you hear this news story? Every single fish they saw at 300 feet was endemic. There's no other place on Earth like that. Every single reef fish at 300 feet on Morrow, and there's a whole story there. What were they doing at 300 feet on Morrow Reef? Every single fish only occurs in the Hawaiian Islands. Pretty cool. Okay, I'm gonna let you go. I know you have to go. Do you have any questions? Let me know. We'll see you on Thursday.